version, you'll also see open. You'll get options for open from BD Research Cloud and save to BD Research Cloud, as well as a new button when adding samples to add samples from BD Research Cloud. So why this exists is because FlowJo many years ago realized that users would save their projects and the FCS files would get disconnected. They'd be saved on hard drives and USB sticks and lost all over the place, and wanted to make a solution to make it a lot easier for immunologists to keep track of everything. So that project went underway, and then they were purchased by BD, where BD said there's a lot more pain points that we can solve via the cloud. And so we wrote down all of those pain points and made the cloud features in FlowJo. But we went ahead and made a website, the companion aspect, what we call the web application for BD Research Cloud. And we said, what do researchers do in a lab? How can we make their jobs easier? And we made workflow features. So you can see here, if you're doing wet lab stuff, you can track the process of collaborating for isolating spleen cells, isolating tumors from salivary glands, isolating islets, like I used to do or be part of at the UF Diabetes Institute here. You could also have these workloads be machine startup. So when you click on here, you can see that somebody's already checked the sheet tank, purged bubbles, pressed the power button, and now they're running in CS&T. So currently these features aren't directly communicating with the cytometer, but that's what I'm working on right now with the team that's making the cytometer agent. So those will go live, but these workflow features already exist, and we're working on integrating them. The heart of BD Research Cloud really is the cytometer. So immunologists use a lot of tools, but cytometers are what BD knows really well. We all use them. They are quirky machines that need a lot of careful QCing. For a lot of cytometry, we don't buy kits. We design panels for these machines, right? So it's very important to know the configuration and have that specific information available to you. So what happens with BD Research Cloud is that Andrea or somebody that runs a core like me would build these configurations and make that information available to you. And then when you do your panel design process, you know exactly which fluorochromes you can excite and exactly which ones you can detect. Now we said to ourselves, well, why are people still designing panels? So somebody, if you've ever designed a flow cytometry panel before, you would know that it's a difficult process. You have to know what reagents are available. You have to know the machine configuration, and you have to know a lot about fluorochromes. Well, why don't we just make this an informatic problem and then be able to build these panels within BD Research Cloud. So I'm going to switch over to this live version real quick. I was on a test version. I didn't realize I had a test that tab open. So I'm going to go back over, or I'm going to stick on my page here, but I'm going to switch to a demo org and not mess up you guys' beta test information area. And I'm going to switch over to Diabetes Institute. There we go. And so I'm going to hit create from this page. And if you want to participate in the beta program, UF is a site. You can talk to Andrea about getting signed up. But if you need to build a panel, you would come here and hit create panel. And let's just call this 8-color PBMC for the sake of just making it easy for this demonstration. I'm going to associate with a project. Everything in BD Research Cloud is in folders. So a project is just a folder of stuff that you're working on. You collaborate on projects with groups of people because science is very collaborative. And then I'm going to head into this panel maker workflow. So like we all know, panels start with the hypothesis really, but you have to choose a cytometer to collect that information from. I could choose from any of the cytometers in the core through this drop-down menu. So let's go with the X20 Magneto here and then complete that step. The next step is where you come in as biologists. So I can do all the panel design thought experiments I want, but it's up to the biologist to know what's going to be important in that tube and how do you define that information. So I'm going to say, let's study humans. Oops. It's a code number there. So I'm going to select that population and we're going to have to fix that bug. I'm not sure how to get past that. Interesting. We pushed a new environment to our production server this morning and having a little bit of a hiccup with that. See if I can't switch back over here and pick up where we left off. So I'm gonna hit create panel, get back to 8-color PBMC, select the project, hit start. Choose a cytometer. And then we're back to our hypothesis step. And you can see here, normally you have all the species that you would know and love. We're going to put in humans. And now we're going to begin the process of building this panel. I'm not sure I can continue because it doesn't look like there we go. So when you're building a panel, back on track here, you have to think about it in the context of populations, really. So you might say my panel includes CD3, CD4, CD8, whatever your favorite markers are, but really those markers are being included so you can identify population. So they'll be mostly positive for things, sometimes negative if there's an exclusion marker within that. So that population definition is important. So I can add antigen targets to this panel, but in BD Research Cloud, what we try to do beforehand is define that population by its targets before we start. And then we can simply add in that population as you go down. So you can see the first population I've added is CD4 T cells. Those are going to be high for CD3, high for CD4, negative for CD8, negative for 14, 16, 19 B cell markers, monocyte markers, and then high for CD45. And then I'm going to come back up here and then type in Tregs and search for them. And then I have a predefined population definition. So if it's not predefined in the system, you can go in and make it. There's nothing stopping you from do that. But if you sat down with Andrea or myself to make a panel, we would say, what in this tube are you looking for? What are targets we can use to identify them? And then you create these definitions and then add them to this panel design process. So similar to CD4 T cells, CD3 Tregs, CD4 Tregs are high for CD3 and CD4, negative for CD8. Low for CD25, right? High for CD45, low, mid probably for CD127, and then positive for FOXP3, also low. I'm also going to mark this as critical because FOXP3 is a pretty critical target for being able to define Tregs. If you ever tried to gate on Tregs where you didn't do good panel design and FOXP3 isn't popping out, it's hard to find. So I'm going to say, because CD127, maybe it's variable, or maybe I don't know what it is on CD4 T cells, I can actually leave that as a question mark. It's kind of an experimental variable. So you don't necessarily have to put in every single one of these targets. The more information, the better. But what we're going to do is take these antigen densities and be able to simulate your cytometer in the expression of these targets on a cell and design panels that will work excellent for you. Likely the first time, but we're going to give you back scores that are going to help you relate this information back to what you're doing. I picked a 9-channel machine the second time around, so we're going to go with 9 floors and 9 channels. I'm going to get rid of that CD127 for this process. So here's where the proprietary stuff comes in that me and mathematicians and developers spent a huge amount of time on. What I'm going to do is I'm going to hit generate panels here and then start talking about what's happening. But there's reference material that we can simulate about your cytometer, about these cells, and the amount of spillover spreading error that will be generated as a consequence of using flow cytometry and the fluorochromes being excited. So if fluorochromes didn't spill over into each other, we could just pick any unique fluorochrome off the shelf. We could pick 9 of them, wouldn't matter, and we could design a good panel. But because we don't know where photons come from all of the time, and that creates spillover spreading error, you have to design panels in a way to minimize the spreading error and maximize the resolution of your target populations. So we can take all of these tables of information. We probably calculated in the order of 1 million different panels that were available from the reagents in the BD catalog and any of the custom reagents that you've created for the system. And we give you back the top 10 panels with a score that is similar to an SSM-like value. So our goal with these scores is going to be a target value of 0, because you want 0 resolution loss and 0 spillover spreading error. But because I designed a 9-color panel on a 9-color machine, we're definitely going to have to make and think about how to put those in different places. So we had a highest scoring panel, which is actually like golf, the lowest score. And you can see that my top 10 panels, we have a 22.18, 22.44, 22.69, 22, 22, 22, 23. All of these are basically in the ballpark each other. And we definitely see that when we design panels, that we probably find 100 panels that all would, if I were to do like a TISNE on this data, you would see that there's a bunch of highly performant panels, and you basically need to make your own decisions on, do I already have these in the fridge? Do I really hate APC size 7 because it falls apart in my fixative, right? So it's up to you to make some of these distinctions at the end of why you would or would not choose some of these. But we spit back out 10. You can visualize results in basically by those SSM-like values where red is going to indicate. And this isn't, I'm working with developers to make a new visualization here. That FOXP3 is actually penalized, and that's why that score is high. It's indicating between panels, really, where you're going to have to make considerations for where that spread is going to land. So if you think CD14, which is just an exclusion marker in this case, is a place where a lot of spread can land, I'll choose this panel, panel 7. So I use this panel, and I can download a report with the actual math, the actual results in an Excel file of how we came across these top 10 panels. And you can look at that later. It's actually saved to this experiment when you come back to it, this workflow, so that'll be available to you then. We don't want to convolute the process with reagents until a later step. So if you've designed panels on the web, they have you choosing reagents right off the bat. It's very overwhelming when there's thousands of choices. We say, these are the fluorochromes, these are the best panels. What size do you want? What clone do you want? So on and so forth. You could restrict by clone early in the process if you so choose, but I could make sure I choose 100 test sizes or however I wanted to do it on these steps. The last step is, because we already know what your panel is, if you've ever sat at a cytometer and tried to label all of the channels. So at FlowJo, I do FlowJo tech support in my spare time. And we know the parameter names, and you know the reagents. So those are the PNS values, the PNS keywords. And when you're sitting in front of Diva and labeling CD3, CD4, and that experimental layout, that cumbersome process, that's what you're actually doing there. But if you already know what the reagents are in a system, there's no reason why you can't just go ahead and make that on the machine. I'm on a development one. I'm actually going to go back over here and demonstrate this within BD Research Cloud. So I'm going to go to experiments, and I'm going to make a new experiment called 8-color PBMC associated with that same project. Choose that cytometer, which, fingers crossed, I think it was the Melody, and our software version, we'll go with that one, and then add an experiment. And then now I can take this experiment and edit it, and I can apply that panel to BD Research Cloud. And so, oops, I got to select it first. So I'm going to select a specimen in tube. I can add more specimens in tubes. And you can see all that information is now being preloaded in the system. A lot of people very commonly will just put the antigen in and make sure that when you get into FlowJo, I don't think we've made this button active on this environment yet, but very commonly you'll just see CD3, CD14, CD19, and that's, the cytometer is ignorant of what reagents you've chosen, right? These are very flexible machines. That's part of the power of flow cytometry, but very often we don't order kits, even though several are available. Most of us are designing these ad hoc. And you have to say to the cytometer, hey, cytometer, I want to run BV510. You know where I'm supposed to be detecting that, because that was pre-programmed in to be that 510 whatever filter off of the violet laser. But it just doesn't know that you want CD3. And you want to make sure in FlowJo, when you have that drop down on the x-axis, that it says CD3 so you can find it later. So we do that here. You can automatically populate it. You can show up to the machine with all of your specimens and tube already made. We have a plate editor as well. You can select your plate size here and set up your plate beforehand. And then it concludes with creating an XML file, an experiment file that it's, the code is XML. And Diva will import that in. You'll get your experiment, you'll get your experimental layout, all your specimens and tubes, and then you show up and start immediately recording. So if you can think about anything that is terrible about the flow cytometry process, we're trying to address those issues. No more, we don't want you using USB sticks, neither does IT. Everything lives in the cloud, everything is available to you. If you go to FlowJo when you're on a family trip and you get bored with they're outside playing beach volleyball and that's not your thing, you go to File, Open for BD Research Cloud. You select your organization. I'm sorry, you log in with your FlowJo portal credentials. And all of that information is going to be saved to the cloud and accessible to you. When you get to, and I'll switch to the appropriate org here so you can see the data that I've uploaded. But Diabetes Institute, autologous cord blood treatment for type 1 diabetes. And we can select a workspace and hit open, and that's all going to be managed now by the cloud. If you're thinking about the process of getting it off the cytometer, like I said, I'm still working with the team that's making the cytometer agent for BD Research Cloud, but currently you would go to our File Explorer page, and it's like just like any other file system where there's folders and subfolders. You're going to select the project files for BD Research Cloud. I'll upload, and let's say I collected some compensation files from this morning. I can go to my desktop, click on my comp folder, hit open. Actually, I got to select them first, right? And then they're going to start uploading, and that's how you get them. It is smart where it knows, it sees these are a bunch of FCS files. You want to make a FlowJo workspace? Sure, let's do it. And then all I have to do is go through the FlowJo application and say, we'll say FlowJo_comps. And then I already have my workspace started. So this project has been ongoing for a little over a couple of years. We want to start automating some of these processes. It would be great if, let's say, you could upload a bunch of files and they would run FlowJo AI, which Christian introduced earlier to the group. And then you knew that all of the samples had been QC'd and should be even analyzed, right? If you have some predefined gating and want to apply that. So we're working on those types of things now. Being a beta test site, we also definitely want your feedback. So in the bottom right corner here, we have a little feedback button. Make sure you don't miss that. You're like, Howie, I want BD Research Cloud to do this. Be a squeaky wheel and you'll get it. You might not get it next week. It might take me a couple months. But my job is to make your life easy. And if you tell me how to do it, then it's possible. Obviously, the squeaky, that's like a group of squeaky wheels gets their things made, but it never hurts to let us know. So with that, I'll say I'm with FlowJo Tech Support a lot of the day. We really love helping customers. Don't struggle through anything. We are professionals. We're here to help. So reach out to us, whether it's for BD Research Cloud, FlowJo, SeatGeek, Rhapsody. We're here to support you. So with that, I'm going to turn it over to Christian, who's going to take it over and teach you more about multiomics. Thank you, Christian. And I'm sharing my screen now. Hopefully you're the co-host still. But if not, we'll figure that out. Sure. Someone shops down. And then, go ahead.
What if there are a lot of unknown entities?
Right. So it's like, how do I input that to the system? And it's basically, if you and I sat down and tried to design a panel that would be good the first try, we could not beat this system. But the system is still not going to know what the energy density is of experimental targets. So you can put them in as unknown, and we handle that as an unknown. So it can be very high, it could be very low. So we maximize the resolution of that target. And if you mark it as critical, we give it extra emphasis to make sure that we resolve that. So it's like, if your RO1 that you're working on, it has that, that gene or that protein target name, like you have to resolve that. And then you might have lesser targets that are still experimental variables that are part of it. And you can put those in as just unknown, and we handle those separately. So there's just the ability to give weighted scoring to those things as you look at it. But a key part of panel design is co-expression, right? And so if CD4 and CD16 are expressed on the same cell, you don't care about how much it's going to show to some degree. So a lot of our testing determined how far can we push that thought process when you're designing panels that are 28 colors to make sure that you can resolve those things still. So there's a lot of that type of, like, I can't anticipate that with my knowledge of flow cytometry, and it's hard to there. Okay, we can talk more at the end here.
Hi, everyone. So I was asked to give a talk on multiomics rather than the high parameter analysis with flow cytometry, but you could, there'll be a talk on high parameter analysis with flow cytometry tomorrow afternoon with Veronica. Today, I'm going to be covering also high dimensional biology analysis, but with multiomics. So an added layer of complexity, but also more comprehensive way of analyzing your data. So why high dimensional biology or HDB? So as you can see here, what I have on my left panel is right here. That's looking at a study that we did internally of NKs and CD8 T cells, where we did dimensionality reduction. So first we generated some PCA followed with some T-SNE. I'm going to go over all those momentarily. But as you can see, it's a little bit of a blob because this is just mRNA sequencing. So with multiomics, what you get is being able to look at more than one omic, an omic being either the proteins or the mRNA. So in high dimensional biology, the idea is to try to acquire the most possible information possible. So what we have here is there's a question, let me see. Could you show slide in full screen? Is that not what you guys are seeing? Oh, they're seeing presenter view. So how do I do the other one? Not that high tech. So if somebody, how do you know how to do that? So this one?
Toggles sometimes. Slides. How do you switch between swap? Yeah, if you hide presenter view, then it'll you could just present and then it'll be one. Is that okay?
Okay, yeah, that's fine. Okay, thank you guys. And sorry, now you guys can see everything. Okay. Oh, and thank you, Chris. You were just typing, Christopher, what I should have done. Thank you. Okay, so that's what we were seeing in the prior, why are you not? Why is it not moving? There we go. Okay, so that's just looking at mRNA sequencing data, looking at gene expression. So for those of you that are doing those type of assays. However, when you incorporate multiomics, so mRNA in terms of what we do at BD, mRNA with protein. So what you get is now this new figure. So what I was showing you is you have this set of islands and clusters that are now very well differentiated when you only look at mRNA. However, when you incorporate protein, now you can see this new portion that I'm putting my cursor on that is exactly telling you how things are dividing and progressing through a trajectory, where what we saw was the least activated being on the bottom left and the most activated being on the top right. So you can see the drastic change you are able to get from your data if you incorporate protein to your RNA sequencing data or RNA sequencing data to your protein data. Otherwise, you're only getting half the story. And when you're doing the analysis, it looks completely different. So why is that? Well, there is a large amount of heterogeneity within every cell. And heterogeneity meaning the number of gene expression, right? So that's the reason why we saw that little blob earlier. On our y-axis in this graph, we see the cumulative TPM, that's transcripts per million. And you can see that on the left, on the right panel figure, we have all the different subsets, and we're seeing all the different contributions individually from each of these subsets to the total heterogeneity of your samples, which is why it's so difficult to get true meaningful results despite having lots of publications making certain assertions. Also, the other problem is that we have low frequency of cells in your rare populations. So these require a lot more enrichment than you would need to do otherwise. But if you incorporate protein and gene with multiomics, then these differences in these rare populations become a lot more noticeable and distinguishable, like I'm showing here. So why would you want to do this? Well, one, your subset enrichment is going to decrease your sequencing costs. So once you're doing, because in multiomics, we're going to be doing it through our platform, which is the Rhapsody, where you're basically sequencing every single cell for number of reads per transcript. But if you enrich, and this is done through labeling and through also sorting, then you're able to reduce those costs and also increase your resolution. So what's an overview of doing some HDB multiomic data analysis? So we start with on the top left, you have a heterogeneous population of cells. Then you load it onto your multiomics platform, in our case being BD Rhapsody. And thereafter, you could incorporate it into any portion of your analysis. So it could either be at the QC level, or you could start at the dimensionality reduction level, or at your gene expression via volcano plots. Now, the reason why we have this circle in the center is because, like I mentioned, you can start at any given time of this portion because it's going to be reiterative. You're going to do your QC, you're going to start doing dimensionality reduction, and all of a sudden you'll see that you need to go back and do more QC. So, or you could do trajectory analysis and all of a sudden decide, I want to look at a particular section of my trajectory analysis and subcluster that, so that we're going back again to clustering or doing another dimensionality reduction. So you're able to toggle between all these phases. So the other portion I mentioned was that, as you can see from the prior figure, let's talk about QC and go around the circle. So we see here that quality control truly matters. Why? You need a high count depth in order for your data to be of actual value, right? Because otherwise you lose confidence in your data. Also, you need to make sure that the viability and the rank of your cells are also of high value. That's why we're doing this, the knee analysis, where anything below that red line, which is where you have the isotope change, that's why we call it the knee, because it looks a little bit like a knee. At that point, you're getting cells that are either dead or dying, or you have empty wells in your analysis. If you don't look for your read depth, your count depth, and remove also dead cells, dying cells, or empty wells, it's going to negatively impact your data analysis. Next portion is about still going with data analysis, I'm sorry, with QC, is the fact of batch correction. So if you're running the same experiment at different runs, you need to remove any batch noise, batch effects. On the blue arrow, we have on the top one where you have not corrected the data to remove batch effects. And you see that it's putting together these two populations, the blue and the brown, as the same cluster, as the same island, maybe two different clusters, but are extremely similar. When in fact, that's not the case. When you remove the batch effects, you can see at the bottom, they're actually, and you can see my cursor, they're actually very different clusters right here. There is an actual separation. And just that alone could lead you to mischaracterize and misphenotype a population, or later on, if you were to bring this back into flow, sort the wrong population. So with HDB, one of the big things is choosing our own adventures. Why? Because once you get data, and data, you get information when you run your multiomic experiment. That gets converted into data when you do your front-end analysis, right? When you convert it into a CSV or something. But then what do you do with that amount of information that is just tremendous? It's not quite well defined in the field. So that's why we have that black box. It's up to you to decide what you want to do with it. Do you want to analyze this via a t-SNE or a TriMap, looking at either at the local or the global architecture? Do you want to do this by traditional means, looking at traditional gating? Or do you want to do basic trajectory analysis, etc.? It's up to you. And that's kind of like what I'm putting on the figure on the right. So these are different type of analyses that it's up to you to choose where you want to go with this. And neither is right or wrong. It just depends on the question you may have. Having said that, though, why do most people in multiomics not just readily do it? That's because most of multiomics data requires a high level of informatics, of prior knowledge of bioinformatics. You're also required to know lots of programming. So having said that, those high barriers of entry, at FlowJo, we developed SeatGeek. SeatGeek is our platform that you use to analyze multiomic data, high dimensional multiomic data, because it doesn't require the programming that I just mentioned. Also, it already follows the best practices for high dimensional data analysis, and it democratizes the analysis to your bench scientists. So most people that are using this, immunologists, now have access to what before was more for geneticists. Also, or that it was limited to bioinformaticians, where if you ran an experiment, you needed to wait months before your bioinformatician got back to you. Now, you're able to get the data and start analyzing it yourself immediately and do the basic and intermediate steps of analysis, freeing up your bioinformaticians to perform the most advanced analyses only. That way, everybody wins. You get your first analysis done, and you free up your bioinformaticians' time to do the more advanced features. Also, SeatGeek is platform agnostic. I've been talking about Rhapsody, but there are other platforms in the market. So whichever platform you use for your multiomic experiment, you're able to load that data into SeatGeek. And also it realizes very much analyses that you do in FlowJo, so the workflow is very similar. Now, how do we analyze some high dimensional data? So I'm going to start as if we were analyzing flow data. Why? Because this is going to be a showcase to your average immunologist, not to your bioinformatician, right? Because a bioinformatician is not going to have need for SeatGeek. Since today's talk is being sponsored by the Flow Core, makes sense that we show how to do multiomic analyses to a cytometrist. So what do we start? Well, in SeatGeek, you have two major views. You have your cell view and you have your gene view. You will notice that under cell view, where I have my cursor, If you're running a multi-omic experiment, meaning that you've already labeled each of the cells with different tags of antibodies, then you're able to gate on the proteins, in this case being your protein of CD4 and CD8. Now you may be wondering, well, Christian, in flow cytometry, I could only label so many biomarkers with antibodies depending on what my analyzer is able to do, how many detectors it has. I'm also limited by my compensation. The one good thing, one of the many good things about multi-omic data is that you don't have to do compensation anymore because these are fluorophores attached to an oligo. So that goes out the window. The second part, you don't have to worry about parameters, again, because they're tagged to an oligo. So you could do as many as possible that is limited by the technology of biomarkers. So you could very easily do 30, 40, 50. And then when you do those, now you have the gene and the protein data. So what are each dot under the cell view? Each dot, as you'd imagine, is a cell. Now, let's now go to gene view. A gene view, every dot is a transcript of a particular gene. So what did we do between the two? All we did was press a button and we could see between protein and genes. And why would we also do gene view? Because with gene view, now you could do gene differential analysis very easily by literally just toggling between the two, gene and cell view, and then selecting the populations of interest if you have already labeled them. In this case, being CD4, CD8 T cells, and over here, we've pre-labeled B cells and T cells, and you can see what genes are upregulated and downregulated accordingly. So what is the usual workflow? You load your CSV, you do your QC and normalized data, like I mentioned already. Normalization, again, is not trivial. And what do I mean with normalization? With normalization, if you're doing a multi-omic experiment, you're supposed to normalize the expression of your proteins, your gene expression to your protein expression. Why? Because genes are expressed on 0 to 10 scale, right? Most likely 1 or 2. Whereas proteins are expressed in the scale of the 10 to the first, all the way 10 to the third, or maybe even 10 to the fourth. So if you don't normalize those expression levels, you're going to get very skewed results when you do especially high dimensional analysis. So that's what I mean with normalization. You have to treat this as, I'm looking at 2 different type of omics, and I'm supposed to normalize the 2 omics into 1. That's one way to look at it. Next, you do your basic gating like I showed you over 4 and 8s, like traditional flow. And then you could pursue further with dimensionality reduction. And then use our plugins for more in-depth analysis. And using these in-depth analysis, then you're able to identify biological processes in novel populations. So I'm gonna go through an experiment that we did and that I analyzed, showing you this workflow. In this experiment, briefly, is we used some mouse model, and we were looking to see whether or not we had differential expression upon treating mice with poly-IC, MCMV, or untreated. So you take and then we use the mouse receptor database that we developed at BV to look for those gene expressions. So we take the mice, collect the cells, then we put it through our sorter and we sort out. And remember, if I bring you back a couple slides beforehand, this is where we said you enrichment. So this is where you enrich to reduce 1, sequencing cost, which is pretty high, and so you could say substantial. 2, you're going to get refined results and more easily identifiable subsets. And 3, you're going to reduce the amount of time and reagents you're going to need to be able to answer your question. So I would highly encourage to do enrichment, unless you're looking at the total differential expression in PBMC. Then for this experiment, then after we sorted the cells, we went into single cell sequencing. And single cell sequencing, we did it with multiomics, right? So we're looking at both RNA and protein. And that's what I'm showing you here. We had 3 groups of mice. Again, those mice with untreated or that were treated with the different conditions that I mentioned. Now you see what I have here on the middle row called SMK, or single cell multiplexing kit, tag 1, 2, and 3. This is another method that you could incorporate into your experiment to, 1, reduce the cost of your multiomic experiment, and 2, facilitate analysis. So what you're doing, in addition to your proteins, you're also labeling your samples with this tag to differentiate between groups. In this case being untreated, MCMV, and our third subset. Pardon, it just skipped my mind. But we had 3 subsets. That's the reason why we get those 3 name tags. So that way we don't have to struggle when we're doing the analysis about what's what. And we could incorporate everything in one cassette. And then we just separate it based on SMKs or use it as part of our analysis. After that, after we added the SMKs, we also added 60 antibodies for phenotyping. So remember when I mentioned that you're limited here, to be able to get 60 antibodies in a standard flow cytometry experiment, it's kind of unheard of right now, right? Right now, what I've seen the most advanced in the literature is 40 biomarkers. And that study, even then, you could see that they were just looking at very high generalities, not very in-depth. Here, you could go as far as you like. We could use already pre-made some panels that we have, like we have this one called the discovery panel. But you could also, if we don't have the tag, we could also conjugate it to your liking. So you're really not limited there. Then we afterwards went ahead, like I said, sorted and then ran this. We loaded the 6 populations that were sorted, and these are the populations, CD8s and NKs, into the Rhapsody. So we're able to do 6 different populations under 3 conditions with 60 antibodies and 3 tags, right? So this is a very complex experiment, but why was this complex? To be able to reduce cost significantly. So again, you'd be asking me, well, Christian, how does this antibody tag, oligo tag work? So we have our antibody, as you can see here. You have your PCR handle that we all know and love. And then you have your antibody specific barcode, which is what you're going to be choosing. Okay. Oops. And then you have your poly A tail. This is why you're able to tag many proteins without worrying about compensation, because there's really no fluorophore there to have an excitation or emission where you would have a bleed over into other channels. So let's go back to QC, right? Because I mentioned that this was highly important. So what I did then is take these samples, well, these data, and then did the need plot analysis. That's my plot on the far left where it says high QC cells. As you can see, I gated all the way into the isotope. I cannot stress enough that your data needs to have high viability, or this is not, or your multiomic experiment is not going to work. So this is one way you can start really seeing how good quality data you have. As you can see in this experiment, we had 98.6% of cells that were of high quality, meaning that they were viable, and they also had a high level of number of genes expressed. That's why we have genes expressed on the y-axis and the rank of the genes on the x-axis. Next, although we did run this on what we call a scanner as part of the Rhapsody to remove that, it removes singlets and doublets. I'm sorry, that removes doublets. I still went ahead and did a little bit of extra work on my own to remove any doublets that may have, snuck in. And, about 4.3% did sneak in, which is really low, but still, we want to remove those. Lastly, I did the last part in looking at my genes, right? Because we're looking at cells. Lastly, we're looking at a QC of genes. What is one thing you need to consider when you're working on this is that you have two types of genes that you really want to get rid of when you're doing your analysis. And this is a point of concern that I have when I see a lot of people doing a lot of work, usually on Seurat. Seurat will give you back an analysis of how much mitochondrial genes you have, but it will not give you how much ribosomal genes or other housekeeping genes, unless you specifically incorporate it into your code. So what does that mean? You may have not the best dimensionality reduction map you would otherwise have, that if you did these steps manually and then ran it through Seurat. So again, this is like kind of being done ad hoc because this is relatively new data, even though it was designated as the technology of the year. So what did I do here? Why did I put that little gate on the top right as potential housekeeping genes? Because what do we know about housekeeping genes? They're highly expressed because they're housekeeping, right? So these include a ribosomal and mitochondrial. So once I look at that box and then I scrutinize it, I notice that I didn't see any in this particular experiment. You may be asking, why is that, Christian? Didn't you just tell me that these might be housekeeping genes? The answer is yes, but the thing that I did not tell you is that I use a targeted panel as well as enriching my cells. Because in a targeted panel, you know exactly what you're looking for. You're not going on a fishing expedition. So you design the primers, or we help you design the primers, so that you only look at the things of interest. If you don't do it this way or with other technologies, when you look at that box on the top right, you will notice that that list that I have, that table, is going to be filled with mitochondrial and ribosomal genes. And then you're going to have to manually remove those from your analysis. In this case, I'm showing you, I don't just have to tell you, but rather I'm showing you the table that if I were to do this just as a run-of-the-mill analysis, I would remove most of my antibodies. So this is why you cannot do this QC steps as a template. Because if you do, you might be removing antibodies of interest, or you might be removing mitochondrial genes. This is something you actually need to go in there and do yourself. The technology is not quite there yet, or at least not in SeatGeek, to do this automatically. And I have not seen this in other softwares either. So this is a point of concern and contention that I would highly advise you to look into with your multiomic data, or just your RNA-seq data, period, before running it through any pipeline like Seurat, or doing analysis yourself. Now, let's go back to, after I said QC, I also talk about normalization. So here, if you click on the button normalization, which is on the top, I don't know if I have the animation, yes. That button on the top is called normalization. Afterwards, there's going to give you that dialog box that I have in the center, okay? The next thing I did is normalize the genes without antibodies. You can see here, I have a total of 329, because again, I did this targeted, right? It's not a WTA, which is a whole transcriptome, where I'm looking at close to 1,000 or maybe 2,000, whatever. I'm very specific knowing what I'm searching for. But again, my gene expression does not correlate with my antibody expression in terms of intensity. So I'm going to go ahead and now normalize the gene expression to the antibody expression. So that's why I highlighted genes without antibodies. And I created two subsets. After that, I'm going to normalize or standardize the antibody, the 54 antibodies that I got here, to, and you saw before that I had 60 because we had some duplicates because we were looking at different epitopes of the same protein because we wanted to ensure we captured it, different clones. So I want to standardize my antibodies to the gene. I'm sorry, my genes to my antibodies. So highlight genes to antibodies, okay? When you do that, this little box over here will change from blue to red. And that will denote that now you have normalization from your genes to your antibodies. Now that we've done QC and normalization, we could go the next step in our grand circle, right? Which is going to do basic gating before we go to clustering. So again, I removed anything that was low viability. So only focusing on my high viability cells, removing any doublets, certifying that my data only contains genes of interest and removing anything that was not genes of interest, followed by traditional flow cytometry gating in my multiomic experiment. So here I'm gating on CD49B against NKG2A. So traditional markers for your NK populations. So I'm going for my double positive. And if, again, we're all used to flow cytometry, this looks very much as if it had come from a flow cytometry experiment, but in reality, it's from your multiomics. The one QC step I did not show you here because it does not apply to this data set is looking at dispersion. Again, like I said, QC is not a template based because dispersion only applies if you're looking at whole data and want to see where differential genes happen, but you're not comparing between two groups. So if I'm comparing, let's say, untreated to MCMB, and I exclude everything that has low dispersion, I'm no longer addressing that question, right? Because most likely all my untreated had low expression of cells, and I removed those. I can't compare something non-existent to something that is existent in the analyses. So the QC step matters what kind of question you're doing. So this is a targeted panel comparing untreated versus two different types of treatments, so I don't include dispersion. If I was doing something else, I just wanted to see the difference of genes within one sample treatment, then I include dispersion QC. Okay, the last part was concatenation. So I showed you a total of eight populations that I sorted from 3 different treatment groups. So I have, if you remember, I had NSMKs and multiple cartridges. So I did also remove batch effects. Once you have all that, once I had all the data from the double positive of CD49B from all those, the double positive from all those cartridges and all those populations, I concatenated. What do I mean with concatenated? I put it all together into one CSV file so I could analyze it jointly. Then we're going to go into the more in-depth, and that brings us into the slew of plugins we have at FlowJo. There's a question. Let me see. Oh, that's it? Okay. Okay. There we go. Yes. So now that I concatenated the file, I want to generate my dimensionality reduction, right? Why? Because you cannot compare 2 different t-SNEs that are generated separately because they're forgetting the word. But every t-SNE differs every time you generate it. So you cannot compare those. Non-deterministic, thank you. Or stochastic. Many words are coming out now. But I first do PCA. Now you may be wondering, how come in flow cytometry, when I'm running a high parameter experiment, I never run PCA, but you're asking me, Christian, to run PCA on a multiomics experiment? The reason is because when t-SNE was developed, it still can't handle a very high number of parameters. It just doesn't do well with that. So you need to go back a step and go basically to its parent of algorithms, which is PCA. PCA is a linear algorithm that does not apply to flow cytometry because it's, again, linear, and data is not linear in flow cytometry. But this is what we have to be able to work with, because PCA is going to collapse your dimensions from hundreds or thousands into potentially between 2 and 25. So it's a lot more manageable than by t-SNE. So you generate PCA, and that's my leftmost panel. And then you take those principal components. And again, principal components could be as many as the number of cells you have. But What I normally recommend is going down and utilizing the principal components up to encapsulating up to 99% of your variance. So anything that contributes less than 1% to variance, I exclude. Okay, so that gives me less than 25 components more often than not. I use those components to then generate t-SNE. And t-SNE, and again, I showed you earlier a different dimensionality reduction map, and that one was TriMap. Why did I do t-SNE now before I showed you TriMap? t-SNE is looking at the global structure. It's looking at the overall and total number of phenotypes present in an experiment. And that's what we get here afterwards, because t-SNE does not give you clusters. t-SNE gives you islands, okay? t-SNE is not a clustering algorithm. It is a dimensionality reduction algorithm. Then I use phenograph to identify the clusters within t-SNE. And that's my rightmost panel, and it's color coded. And you can see how gorgeous these clusters are. I mean, right? They're not like spread all over the place. They're not overlapping with each other. They're exactly where they need to be. So then I looked at the SM case, which is my 3 different conditions, untreated or basal, my MCMB, and my poly-IC. And I asked in a question, do my cells that are treated for MCMV and poly-IC, i.e. my treated groups, do they cluster? Because again, this is a type of a QC question, right? Because if they were spread all over the place, what do I know? Something went wrong. But what do we see if we're looking at the global architecture? We see that all the phenotypes that are activated, what do you know? They're all on the right hemisphere of my t-SNE map. So I would recommend that even when you're doing analyses, you're still doing QC. So analysis and QC go hand in hand in multiomics data. Because if it doesn't make sense, there's something wrong. Okay, but I picked up these clusters. They make sense, sort of. I mean, why do I say sort of? Because I don't know if these clusters are actually of high QC. All I know, they're about in the right location, but I still need to evaluate those clusters. So that's where it takes us to one of our plugins called Euclid. In Euclid, it gives you 2 different visualizations. This is one of the 2. In this particular visualization, you're comparing 2 clusters and you're seeing the difference between the 2. The higher they are in the legend, the more different they are. That's variability. And you could set that variability to whatever you want. In this example, I set it to 0.08 because I was being extremely stringent. However, the standard is usually setting it to 0.5. And if they're very different, they'll be closer to 0.5. I was just being extremely stringent because I really wanted to look at the very rare populations here. So what am I looking at here? Well, if you notice, cluster 9 and 20. Oops. Oh, I didn't get my little circles. Cluster 9 and 20. So let's go cluster 9. Boop, boop, boop. 9 and 20. Here we go. You can see that's very light in color, meaning that that cluster is extremely, the cursor goes away. Here we go. The cluster is very different. However, excuse me, cluster 7 against cluster 11, which is right here, is very dark, meaning that those 2 clusters are very similar. Why is that important? And this is where being an immunologist is key in your multiomics experiment. If you gave this to a bioinformatician that's exclusively into bioinformatics, he'll know there's a difference, right? He could detect that. However, is that difference biologically significant? Because that is the real question as immunologists we ask. Is this even relevant, or should I not care? Let's say you had an expression level of 1,000 of CD3, and you had another group of cells that had 990. Bioinformatically, they're different by a figure of 10, right? Biologically, does that matter? No, because you're 990 and 1,000. If I was going to create a gate, I'm going to gate them all together. So that is what I mean with being also looking at your data critically, but also thinking about what you know about immunology. A bioinformatician is going to give you and tell you, hey, Christian, you have 24 clusters. The truth is that once I go through this, not all those 24 clusters are significant biologically. I'm actually going to end up with something like around 16 when I actually did this for the real analysis. So I would highly encourage you to do that. Also, if you don't incorporate multiomics, you're not going to see that big of a difference. Why? Because it has been shown in the literature that there is a obvious discordance between gene and protein expression. So while you may not always get expression of a particular gene, that does not mean that the protein is not being expressed. So if the protein is expressed because it's stable, but you get no transcripts being done, you could miss that phenotype because it will come back as negative for your gene expression. This is why doing multiomics is so critical. The hand in hand goes also with doing only flow. If you only do flow, you don't know what's happening at the gene level. We know of different viruses or different conditions that will silence a gene, but if the protein is stable, whatever was already on there, it's already on there. And like anything, every experiment we do is a snapshot. So if you didn't catch at the right time, you may have protein, but you're not seeing what the virus or what your condition might be doing at the gene level. So you want a real story, a complete story, not a real story, but if you want a complete story, do multiomics. And the more omics, the better, right? BD, we could only handle 2 at the time, right now, but multiomics goes beyond that. It goes into metabolomics, probably pronouncing it wrong. And there's a lot of more omics. And as technology progresses, we'll be able to incorporate more and more data into it. Again, as you saw, I showed you that we could incorporate, and you could start at any of these steps and go back and forth. So I could have gone into QC and like I showed you like a linear progression. But once I got to a cluster, I could go backwards again and evaluate the QC of that cluster and then start that circle again. So with a multiomics experiment, the other beautiful thing about it is you could spend hours and hours on it and days and get several publications out of it because the data is so rich, provided that the experiment was well planned out and the QC is done well. Now, here, oh, now my box is here. Then the other one is cluster 1 and 9. So I'm going to show you this comparisons that I did. I remember when I was talking about biologically relevant or only bioinformatically different. Look at cluster 1, look at cluster 9. Cluster 9 is on the left, cluster 1 is on the 9 is on the left, 1 is on the right. Those that are labeled AB at the end are protein expression. Those that do not have AB are your genes. So for instance, we have a high level of IL-2 being expressed in both, but we have that both of them are negative basically for CD69 protein. If you look at this, would you say that these are different? Probably not. This is what I mean that and not everything that clusters out informatically is biologically relevant. I would actually merge these 2 clusters together and only make 1 cluster. And I would challenge someone to tell me that I couldn't do that. So when we get data from other people analyzed, we need to ask ourselves, are these data making sense? Do these data mean anything biologically, or is this just an artifact from mathematics? So please take the time to really scrutinize your own data. Now, what about TriMap? I talked about TSNE, TriMap. So TriMap looks at the global structure, at the local structure, pardon me, the global structure, TZ was local structure. So the global structure, you want to start looking at your trajectory. I showed you at the beginning of my talk that you had like this blob when you didn't incorporate proteins. You had only genes. This one, I'm incorporating proteins. So this is without clustering. So I just get this contour mapping with TriMap. It's showing me like these little fingers, right? Well, what do I know about it? Not much, but I know something interesting is happening there. Then I ran clustering with, in this case, being XShift. I recommend usually running XShift with TriMap and Phenograph with TSNE. And if you like, I can send you some publications why that's the case. But generally speaking, XShift runs a lot faster. It's a much more modern, what do you call it, algorithm, but it takes more into account the nuances that TriMap does rather than Phenograph. So here, when I did this and I start scrutinizing my different clusters, what I found out is that you go from lowest level of activation to the highest level of activation, following those different areas. This you could not see with TSNE. So why did I run TSNE? First, to see what are all my phenotypes here? What do I have? The next thing I ran was a TriMap analysis. Why? Because I want to know, okay, I have these phenotypes, but what do they do? Right? Otherwise, it'll be too difficult for me to try to put together a story. And the main story usually is my cells change from this to this, from non-activated to highly activated, from non-degranulated to degranulated, or from a stem cell to a CD8 T cell, etc. There are different questions, different algorithms for different questions. Yes. Here we go. How to tell the direction. So that one we have to do an analysis into each of the major clusters. So let me show you how I did that. That's essentially it. I then generated a heat map, and this is using our plugin called Violin Box. And what I did was associate the different clusters here, because actually will give me numbers, and then I correlated them to their phenotype. And based on their phenotype from this heat map, I'm able then to go back to my Phenograph and see, to my TriMap and see that the cells at the bottom left are non-activated all the way to the top, which are very activated. And if we see here, we also have dendrograms with our heat map. They're basically breaking up into two major subsets. This is one subset, my high levels expression of CD154, which is associated with activation, remember correctly. And then followed by these two other major, and then followed into one major subset that gets further broken into two. This region here, and then this whole region, and that gets further broken up into two. Okay. With Violin Box, you can also generate dendrograms on your x-axis. And I'm not showing you on this slide, but then you're able to start grouping the clusters together. And what I saw in my heat map analysis is that the clusters from the bottom left were part of the same group from the dendrogram analysis. So all of these starts coming together again from the circle that I was mentioning. You start going back and forth between your analysis, your dimensionality reduction, your clustering. So you could start at any one part and always end up with more information. So what do we know? Then I think I demonstrated that if you only use genes or all proteins, you only get half a story. And I showed you with relevant actual experiment data, some wet lab experiment. But when you incorporate proteins to your multiomic experiment, you're now only able to tell a story about trajectory. If I had only done mRNA, odds are I wouldn't be able to tell you that, or not with high enough confidence. Here, I have the whole story to lay at you and show you this is what is happening both at the protein and at the gene level. And that's all I have right now. I hope this made sense and why multiomics is very important and the workflow that we do to be able to generate these data. And these are just my acknowledgements of the different colleagues from BD that helped this study possible. And one more question. What's the cost for one sample? I'm not sure what you mean, Dan. Cost per cartridge or cost for sequencing, overall cost? That, again, is going to depend based on your experiment. Unfortunately, there's not like flow cytometry where we know how much antibody there is. You could run an experiment and you could ask Andrea what the cost from the core is to run the experiments, and she'll let you know. She'll help you with all of that. Sequencing, I don't know if UF does the sequencing on site or not. Andrea's telling me that you guys do. So please go to your core. Yes, come talk to Andrea. She'll fill you in. And if you guys need any help, we're also here, all at BD, here to help, to provide you with analysis support if you need any analysis support. I want to see if there's any other questions. I don't think there is. All right, well, I appreciate you guys' time. Thank you so much. And the recording will be made available again at the core facility. And I also want to again thank everyone at the core for making today possible, bringing us out, inviting us, and being the generous host that everyone here at the core is. And I guess we'll see you guys next time. Thank you so much. Thank you.